Good evening, everyone. These are going to be the next notes over the fermentation systems that professor gave us to study for the next exam. All right, so looking at the systems of fermentation, we need to be taking into consideration for the media that we're going to be using. We have media costs to be worried about. We have to look at the material, we have to look at the product yield, the availability of those said materials, and the transportation costs. Carbon sources, so carbohydrates, again, the difference between certain carbohydrates could be very important in the fermentation sense of things. So pure glucose or sucrose are seldom used because they're actually expensive. That's why I have here not costly glucose or sucrose. Instead, we could use things like molasses. This is like that syrupy stuff. This is a cheap source of carbohydrates, such as sucrose. Sucrose is found in molasses. So are nitrogen and vitamins. You could have different composition of molasses, so it'll vary on the source. So you could have beet or cane molasses, and then it's also going to vary on the climate. The other option as a carbon source you could use is malt extract. This is from malted barley. Sorry about that, I have a cold, I just need to grab a tissue. So malt extract is the second choice for carbs. And it will have glucose, fructose, maltose, and sucrose. So we have 4 types of carbohydrates in this extract. Nitrogen will be from the proteins and the peptides, and when we're heating malt extract, this produces the Maillard reaction, and this will create these products between the amino groups and the reducing sugars. Therefore, you can't use this Maillard reaction product. You can't use that as a nutrient, so therefore you can't really heat the malt extract. We have 4 other sources of carbs. We have starch and dextrin. These can be used by microbes that have amylases, so we need some amylase microbes such as bacillus, and they could also be converted into a glucose syrup and then used as a substrate. That's for starch. We also have sulfite waste liquor from the paper industry. And there's going to be 2 or 3% glucose, mannose, and galactose in the sulfite waste liquor. Difference here, galactose kind of shows up here. You do have some, never mind, I misread that. So just know that in the sulfite waste liquor, we have 2 or 3% glucose, mannose, and galactose. Cellulose, we could get that from straw, corn cobs, or wood waste. We would need a chemical or an enzyme to break it down into fermentable sugars. Whey contains a lot of lactose as well as proteins, and however, the storage and the transport costs make it uneconomical. It's too expensive to transport whey. It's also too expensive to store it, so that's kind of like a last resort, kind of. Alright, nitrogen sources. We could have ammonium salts, urea, and gaseous ammonia, or NH3. We're going to find nitrogen in corn steep liquor, where we're going to have the autolyzed baker's yeast also as a source, and then peptones. So let's go back to corn steep liquor. This is a 4% nitrogen from cornstarch production. This corn steep liquor is a product of cornstarch production, and it has this 4% nitrogen content. Yeast extract, again from autolyzed baker's yeast. This will contain amino acids and vitamins. Peptones are going to be hydrolyzed meat, casein, gelatin, keratin, peanuts, and soy meal. And you're going to want to feed the amino acids, not difficult proteins. So this is again going to be a source of amino acids which could be used for their nitrogen content. Also want to look at the pH and the temperature of the solution So pH-wise, you want a pH of 4 to 8 for most bacteria. Acidophiles are going to be a lower pH, and alkylophiles are going to be a higher pH, because they like that basic condition versus the acidophiles, which of course like acidic conditions. The best pH for growth doesn't necessarily mean the best product pH or the best pH for product formation. You could have small changes in the pH, but these could have dramatic or huge impacts. So you need a controlled pH. Likewise with temperature. So temperature, you can have thermophiles, which are going to be grown from 55 to 120, but they could also be isolated at 300 degrees Celsius. Psychrophiles are going to be at the polar regions. That's going to be less than 10 Celsius. And then our mesophiles are going to be 20 to 40 degrees Celsius. This is where you find some of those pathogens as well. You could use feedback control via temperature probes or insert it into the fermenter to monitor and control that temperature for the media. Cooling is usually only possible. The heat comes from the reactions. Going to also want to take into consideration foam. So foam will be created through the impellers, the addition of gas, and the growth of the cells. You're going to want to control dissolved oxygen, and at the same time, these same strategies are going to also control the foam. You could break down foam using antifoam, which affects the metabolism, unfortunately. You're going to use antifoam to lower the surface tension. Or you could use a foam breaker, which is a mechanical object mounted onto the stirrer above the liquid. Alright, let's talk about different batch systems. So we're going to start with the regular batch, and then we're going to go to the fed batch. Okay, so the batch system. Batch fermentation is a closed system, as we have in this first part here. There's going to be no additional nutrition other than the start at time equals 0. Nothing will be added. The only things you're adding will be things to change the pH. For control, you're also going to be adding some oxygen and antifoam. So these three things. You're not playing around with the nutrients at all. You're going to have several phases in batch fermentation you need to watch out for. Our first one, where the kind of like a trapezoidal parabola, I guess that's how you could describe it, that kind of dips down into perfect actual parabola that's opening upwards at the end. So at the very bottom we have our lag phase. The lag phase is where there isn't an increase in cell numbers. They're adjusting to pH, and they're going to be synthesizing the transport systems. And the lag phase, it depends on the phase of growth of the inoculum. If a culture is in log phase, there won't be any lag. Log phase is also known as exponential phase, so this is where we're actually going to see the doubling of cell number versus unit of time in yeast and bacteria, whereas the doubling of cell number biomass, or the filament in the system, will be in fungi. For simplicity, I think I'm going to just call this the exponential phase, so we're not getting confused between lag and log. So this is going to be a straight line when cell numbers or biomass are plotted on a semi-log graph. It's also independent of substrate concentration as long as the excess nutrients are present. The growth of cell mass is described by a doubling of cell number per unit of time. In the exponential phase, we could use the Monod equation. It's based off the MM, or Michaelis-Menten equation. So we have this U equals UM multiplied by S divided by, in parentheses, KS plus S, Okay, so let's break down all of those variables. So this first U here, I think that's a mu. I can't remember my Greek alphabet, but it's that U that has like a long pair of tails. So that is going to be the specific growth rate of the microorganism. is going to be the max growth rate. S is going to be the concentration of the limiting substrate for growth. Ks is also known as the substrate-specific growth rate. The substrate concentration when half the specific growth rate is obtained, aka the value of S when U over U max is 0.5. Alright, so let's write that out. So Ks is the substrate-specific growth rate. And then again, this is going to be the substrate concentration. So we're going to put that in those hard brackets for substrate concentration when 1/2 of the specific growth rate is obtained. Remember, our specific growth rate is mu. In other words, the value of S when mu over mu max is 0.5. The Michaelis-Menten for log phase, aka the specific growth rate or the Monod equation, it relates growth rate to the limiting nutrient. In the exponential phase, we also have something called dioxic growth. And so in this, glucose is preferably taken in by the bacteria. The uptake of glucose will repress the uptake of other substrates, such as other carbs. Once glucose is exhausted, the second substrate is taken up. So for example, when we use up all the glucose, maybe the bacteria will take up all the lactose. It will start making that, the lac operon and getting all those enzymes there for it to use lactose. There's some examples here. Glucose plus xylose or ribose. You don't want another lag. You don't want another phase where there's no change in the number of cells. So you would want to use something like glucose plus mannose, because there isn't a lag, nor is it dioxic, so we won't see any dioxic growth when we have a mixture of glucose plus mannose. The stationary phase is after the exponential phase. Stationary because it basically plateaus here. So a growth slows then stops when a critical nutrient is depleted. So we ran out of a nutrient, or there's a ton of toxins in our fermenter. The death and lysis of cells could provide nutrients to their survivors. So this idea of you're built upon those who are previously around, that's kind of the case at stationary phase. The death and the lysis cells will provide nutrients to survivors. We can make secondary metabolites in the stationary phase, whereas primary metabolites are actually made in the exponential phase. We also have the death phase, but then let's move on to the fed batch fermentation. As its name suggests, you're going to be adding the substrate in increments throughout the duration. Instead of adding all the substrate at the beginning, you're going to, again, space it out in these equal amounts. Fed batch is used in the penicillin industry. It prevents catabolite repression by high concentrations of glucose or other carbohydrate or nitrogen resources. It's not usually possible to measure the substrate concentration. However, you could use indirect parameters. Can be correlated with the metabolism of the critical substrate. For example, the production of organic acids, pH could be used to adjust feed rates. Let's talk about the continuous fermentation. So first off, we're going to start with the homogeneously mixed bioreactor. This is a continuous and an open system. We have two types. We have the chemostat, which is going to be in a steady state. The cell growth is controlled by adjusting the concentration of one substrate only. However, in the turbidostat, turbidity is monitored and used to keep the cell growth constant by adjusting flow of the medium. So in chemostat, you're changing the concentration of one substrate in a steady state of cell growth. However, in the turbidostat, as the name implies, turbidity is being monitored, and that keeps the cell growth constant by adjusting the flow of medium. We also have the plug flow reactor. This is another continuous fermentation system. And the nutrient solution will flow through a reactor without an agitator. We don't have an agitator here. The cell density will vary in different parts of the reactor, however. At the inlet, the nutrients will be added continuously. We have three types of fermentation processes. Type 1 is when the product is derived directly from the primary metabolism. So basically, A to product, or A to B to C. The product could be biomass, it could be the single cell peptide, it could also be ethanol. This product is used in energy production since it's derived directly from primary metabolism. I'm going to write our little chart here, A to B to C, or A to product. That also works too. Okay, type 2, let's see its difference to type 1. Type 2 fermentation, the substrate is still used for primary energy production. However, the product is produced by a secondary pathway which is separated from the primary. Okay, so an example of this would be our A, B, C. However, A could also go to E, and then that could go to our product. So up here, the product was C in type 1. However, we could either go to A or A to B or A to E. That's like a branching point in the energy production. So if we have that branch there, or we have a choice of A to B or A to E, and then A to E goes to our product, that's going to be type 2 fermentation. Examples would be citric acid and some amino acids. Type 3, this is when the primary metabolism and the product formation occur at completely separate times. They're completely separated. Primary metabolism goes first, and then substrate consumption and growth. Product is formed by the reactions of intermediate metabolism. This is going to be found in the antibiotics and the vitamins. And then we also have two equations here for productivity. Productivity P in units per hour would be equal to the product concentration per liter over fermentation time. We could also have the yield coefficient, which is YS. That's going to be the biomass in grams over the substrate consumption in grams as well. So two handy equations at the end for productivity. All right, that's going to conclude part 1. Please stay tuned for fermentation part 2. Please do something nice for someone and take care.